Good day, everyone. 안녕하세요. This is the only Korean that I learned and understand. Anyway, it's a great honor to be invited on this webinar series to give CRISPR technology that offered by GenScript to the Korean audience. So for the past 10 years, the genome editing has been developed at a fast pace, especially since the discovery of the CRISPR system. So the maturations of the CRISPR system has been utilized in various applications, ranging from the basic research to the applied biotechnology. So today, I am going to share with you how we harness this technology for different applications and what GenScript could have offered to help our researchers in their work. So let me guide you through to the content of my presentation today. So I will first give a brief introduction about the CRISPR system, and then followed by how the RNP system could have helped to accelerate and simplify the entire genome editing workflow. And lastly, I will wrap up my presentations by presenting some of the extended product that extended by GenScript based on the CRISPR technology. So just a little bit of the background here. In mid-2000, the researchers have found that many of the bacteria consisted of the repeated sequences, as shown in the black diamonds here, that flanked with the unique sequences, as shown in the colored boxes in the genome, which is called the CRISPR, which is also an acronym of the clusters of regularly interspaced short palindromic repeats. The researchers found that these repeated sequences, these unique sequences are the DNA that come from the virus. So the researchers also identified the CRISPR-associated or the Cas gene. And these Cas genes were consistently adjacent to this CRISPR loci and presented the motif characterizations of both the helicases and also the endonucleases. Additionally, the researchers had also discovered the tracer RNA that nearby the Cas gene. So here I show you a simplified workflow of how the CRISPR system in the bacteria. So the CRISPR-Cas adaptive immune response in the bacteria consists of two phases, the immunization phase and an immunity phase. So in immunization, the Cas protein will form a Cas complex that will recognize the genetic materials from the virus, whether it's a DNA or the RNA. And then it will recognize a specific regions of these genetic materials and cleave these genetic materials into pieces. And this piece of DNA will then be incorporated into the CRISPR array and form a repeat and spacer unit. So in the immunity phase, so this repeat and spacer unit will be transcribed into a SERNA precursor. So the Cas complex will be guided under the tracer RNA, and then to bind with the SERNA to form a SERNA tracer RNA Cas complex. So this Cas complex, the maturations of this complex will be cleaved under the RNase-3 DNA, RNase-3 polymerase. So this RNA will play a role as a guide RNA that will recognize a specific region in the viral DNA or sometimes even the RNA, and then will track the Cas protein into these specific regions and trigger its cutting ability to cleave the genetic materials into pieces. Okay, so this is really the step that we could actually harness for genome editing technology. Okay, so through the study, the researcher discovered that the Cas protein can actually recognize double-stranded DNA, and then where the positions, it will actually match with the 20 nucleotides of the guide RNA. So there are several types of the Cas endonucleases, but only the Cas9 endonucleases is commonly used in the market for genome editing. Also, another thing that the guide RNA that I show here is a single format in which the 20 nucleotide of the rRNA and a 67 nucleotide of the tracer RNAs are covalently linked together. So this is important so that it allows the researchers to design the desired sequence that can recognize a specific regions in the genome that can track the Cas protein to the specific site of the genome and then trigger its cutting ability to cut the double strand, the DNA into pieces. So the double-stranded break can be repaired through two different pathways, a non-homologous embryonic pathway and a homology-directed repair pathway. So in brief, when there is no DNA donor template, the repair will be carried out through an error-prone non-homologous embryonic pathway in which the random nucleotides around the breaks will be deleted, resulting in an indels that cause the frame shift, which subsequently will lead to gene knockout events. In contrast, when there is a DNA donor template, which flanked with a long homology arm at both ends of the fragment in the cell, what the cell will do is to integrate these fragments into one strand of the genome because the homology is there. And then the sequence in the middle will be repaired to another strand of in the genome. So this in order to create a gene knock-in event. So this is how CRISPR can be utilized, whether it's to create a gene knockout or a gene knock-in. So with the understanding of the CRISPR system, it enables the researchers to have a better genetic or epigenetic controls, and potentially can be broadly used in different applications, as I mentioned earlier, from the basic research to applied biotechnology, such as the alteration of the functional, the biological functions, and then also to change the disease phenotype, to introduce the mutations into the cells, and then to create a synthetic materials that are useful. In food industry, it can also be utilized to improve the crop, such as resistant to the pathogenic infections, and also to improve the metabolic pathway for fuel production, or to provide solutions for the gene surgery and drug development. So the CRISPR delivery systems can be discussed from two different perspectives: A, the cargo, and B, the delivery vehicles. The three approaches of the CRISPR-Cas9 cargos are the DNA plasmid that encode the Cas9 proteins and the guide RNA, the mRNA for the Cas9 translation, alongside with a separate guide RNA that is a dual oligo format of the guide RNA, and then a Cas9 protein with a guide RNA, which is a ribonucleoprotein complex, or we call it a RNP system. So this table summarizes the pros and cons of three different cargos. The plasmid system consists of two different plasmid systems, an all-in-one expression system that encodes both guide RNA and Cas9 simultaneously, and a dual plasmid system that encodes the guide RNA and Cas9 separately. The advantages of using these two plasmid systems are easy to use, renewable, and low cost. But the studies have shown that the use of these plasmid systems are less efficient and higher off-target rates. The only difference between these two plasmid systems is some of the studies shows that the use of the dual plasmid system may have improved the editing efficiency and efficacy in some of the cancer cell lines. So the second cargo involved the use of the mRNA for Cas9 translation and also a dual oligo format of the guide RNA. So the advantages of these cargoes are DNA-free and bypass the transcription because the use of the mRNA directly for Cas9 translation, and then faster editing and degradation, minimum off-target rates, high editing efficiency. But the drawbacks of these cargoes is a higher cost, and because of the use of a foreign mRNA for Cas9 translation in the cell, it may result in the immunogenicity or the cytotoxicity to the cell. So the advantages of using the RNP systems are DNA-free, bypass transcription and translation because the use of Cas9 endonuclease directly, fastest editing action and degradation, minimum off-target rates, highest editing efficiency, and most importantly, the studies have shown that it has the lowest cytotoxicity effect with the use of the RNP system. The only drawbacks for this RNP system is higher cost compared to the previous two cargo. So in the later of my talk, I will focus more in the RNP system because this is a system that can really help to accelerate and simplify the entire genome editing workflow. Now, the CRISPR-Cas9 can be delivered through two different approaches, either the use of a non-viral based delivery approach and a viral-based delivery approach. So the non-viral based delivery approach relies on either the chemical or a physical mechanism. The chemical mechanisms relies on the use of a lipid-mediated transfections that involves the use of a cation lipids or cation polymers. And they have to form the liposomes between a negatively charged nucleic acid and a cation polymer lipids. And this liposome will then merge with the cell membrane and release its content into the cytoplasm. Or relies on the use of a calcium phosphate transfections approach by mixing the DNA with calcium phosphate in order to form the condensed calcium phosphate DNA precipitate. And this calcium phosphate can help to bind to the cell surface and allows the DNA to get into the cell by endocytosis. The physical mechanism relies on the use of an electroporation approach that is to use the electric pulse to physically cause a temporary pause into the cell membrane to allow the DNA to get into the cell. So here I show you the advantages and the disadvantages of all of these approaches, but due to the time constraint, I'm not going into details. So the viral-based delivery method relies on the use of a lentivirus, an adenovirus, and the AAV vector. Well, the viral-based delivery method is commonly used due to its high transduction and editing efficiency. So, but the studies have shown that the use of the viral-based delivery method may have caused the high off-target rates, and also its ability to integrate the virus genome into the host genome and cause the onset disease raised concerns among the researchers. Additionally, the use of a viral-based delivery approach requires a viral packaging step, which could be a pain point to some of the laboratories that do not have an equivalent setup to handle the viral packaging. So now I would like to switch gear a little bit to talk about how the RNP system could have helped to accelerate and simplify the entire genome editing workflow. Here I show you an article that published by Kim et al. From Seoul University in 2014, in which the author studied the RNP system to improve the genome editing efficiency and efficacy. So in this article, the author compared the RNP system with the plasmid system in the hard-to-transfect human cell line and to measure the mediated mutations using the T7E1 assay. So as you can see from these results here, the RNP system could induce an indels at a higher frequency at 23% compared to the 10% in the cells were transfected with the plasmid. Furthermore, they had also studied the cell viability. So in this study, it shows that the number of colonies that transfected using the RNP system, which was shown in the red bars, were at least twofold higher compared to the number of colonies that transfected using the plasmid system, indicating the lower cytotoxicity with the use of the RNP system. So to investigate whether or not the RNP system had lowered the off-target rate, what the author did was to select 3 different sites where previously were shown to induce the off-target mutations at a higher frequency and compare it with the plasmid system. So as you can see from this result, the RNP was highly active and induced the indels at a frequency ranging from 76% to 79%. And most importantly, is the RNP system could reduce the off-target rate significantly compared to the use of the plasmid system. The author had also conducted the time course study in order to investigate the kinetic of the RNP system and then to measure the indels using the T7E1 assay, also the Western blot analysis. So you can see that the RNP systems started to cleave the chromosome DNA almost immediately after the transfection and induce the indels induce the indels reach its plateaus at 24 hours after the transfections, whereas it took about 3 days for to reach an equivalent level of the mutations rates with the use of the plasmid system. So the Western blot analysis showed that the Cas9 endonuclease that expressed with the use of the RNP system was degraded rapidly in the cell. So as you can see from this Western blot analysis, so after the 24 hours of the transfections, the Cas9 endonuclease was barely detected, whereas the Cas9 endonuclease was expressed from the plasmid system for more than 3 days. This could potentially lead to the accumulations of the off-target mutations in the cell. So in summary, the advantages of using the RNP systems are faster editing results, readily degraded Cas9 proteins, as well as less off-target cuts. So one thing I would like to highlight here is this study was conducted in the year of 2014, in which the technology to chemically synthesize the sgRNA, a long nucleotide of the sgRNA was not available. And therefore, the sgRNA that was used in this study was in vitro transcribed using the T7 RNA polymerase, which was known as the IVT sgRNA. A recent study by the same group of the researchers showed that the use of the IVT sgRNA may have brought the immunogenicity or the cytotoxicity issue due to the presence of the 5' triphosphate moiety in the sgRNA. So it was hypothesized that the 5' triphosphate will make the sgRNA to be recognized as a foreign or a non-self RNA that will activate or induce the type 1 interferons, and which intends to activate the antiviral effector proteins, including the DDX58 or the OAS2. So to prove that, what the author did was to incubate the CD4 plus cells with 2 different types of the sgRNA. That one consists of the 5' triphosphate, and another one was treated with the seed in order to remove the 5' triphosphate, resulting in the presence of the hydroxyl group at the 5' ends of the sgRNA. So the results show that the mRNA level of the DDX58, the OAS2, and the type 1 interferons were highly increased or expressed in the CD4 plus that transfected with the sgRNA that consists of the 5' triphosphate, indicating the inductions of the innate immune response that could lead to the cytotoxicity. So all of this information comes to the conclusion that it is important to have an sgRNA that without the 5' triphosphate in order to improve the genome editing efficiency and efficacy as a result of avoiding the induction of the innate immune response. And also, during the time where the technology to chemically synthesize sgRNA was not available, a dual oligo format of the guide RNA was that consists of 20 nucleotides of the SERNA and 67 nucleotides of the tracer RNA was provided. So, with this dual oligo format of the guide RNA, what we need to do is to anneal these two strands first before transfected into the cell. But the studies have shown that these two strands may dissociate easily. This could lead to the poor stability when duplex with the Cas9 endonuclease, resulting in the poor editing efficiency. Fortunately, with our in-house technology, GenScript managed to chemically synthesize a long nucleotide of the sgRNA currently. And unlike the dual oligo format of the guide RNA, the annealing is not necessary anymore, and therefore it will be more stable when duplexed with the Cas9 endonuclease, resulting in a higher editing efficiency. So, GenScript had also performed a comparison study to investigate the editing efficiency between the use of the sgRNA and the dual oligo format of the guide RNA. So in this study, both of these two different types of the guide RNA were modified at the first three 5' and 3' terminal RNA residues in order to ensure a better stability. So this gel photo shows that at a similar concentration, the sgRNA can induce an index at a higher frequency at 82% compared to the 64% with the dual oligo format of the guide RNA. Furthermore, GenScript had also investigated how the purification method could have helped in terms of the editing efficiency. So the gel photo showed that the sgRNA that purified using the HPLC could induce a higher index frequency at 76% compared to the 66% with the sgRNA that purified with the dissolved method. So in the conclusions, the sgRNA could have performed better than the dual oligo format of the guide RNA, and the further higher purity could have boosted the editing efficiency. So this table summarizes the advantages and disadvantages of these three different types of the guide RNA that I mentioned. The IVT sgRNA requires the in vitro transcription and the purification step. And as I mentioned earlier, this IVT sgRNA may have brought the immunogenicity issue due to the presence of the 5' triphosphate. And the chemically synthesized of the sgRNA, which without the 5' triphosphate, is a readily used sgRNA, and it can be easily modified to make it more stable in order to improve the genome editing efficiency. And most importantly is this sgRNA has a low immunogenicity and cytotoxicity effect. The dual oligo format of the guide RNA requires a multiple step to anneal the SERNA and tracer RNA. And as I mentioned, these two strands may dissociate easily, resulting in the poor stability when duplexed with the Cas9 endonuclease, resulting in the poor editing efficiency. Right. So here I show you the workflow of how RNP system could have accelerated and simplified the entire gene knockout workflow by taking the T cell as an example. So what you need to do is to design a guide RNA. So and then with some modifications in order to make it stable, and then to mix it with the Cas9 endonuclease for 1 hour in order to form the RNP. And then while waiting, the T cells can be isolated and resuspended in the electroporation buffer. So after 1 hour, the newly formed of the RNP system can then mix it with the cells in the electroporation buffer and to further electroporate for 30 minutes, followed by the cell expansion in the 96-well plate or for downstream analysis. So currently, GenScript offered two different types of the sgRNA, the easy edit sgRNA and a safe edit sgRNA. So both sgRNA type could achieve a similar editing efficiency. The only difference is the safe edit sgRNA has a higher purity to ensure the minimum cytotoxicity effect and off-target rate, especially to the project that involves the primary cells and stem cells. So that's all for the CRISPR knockout. Now I am going to talk about the CRISPR knock-in using the DNA donor template. So conventionally, the viral vectors is used in order to deliver the DNA as a donor template due to its high transductions and editing efficiency. But as I mentioned earlier, the use of the viral vectors may have the high off-target rate issues and also its ability to integrate its genome into the host genome may cause the onset disease. That is also the main concerns among the researchers. But thanks to the maturations of the CRISPR technology, it is now possible to deliver a large gene site into a specific site in the genome without the use of the viral vectors. And this leads to the potentially use of a double-strand DNA or a single-strand DNA directly as a donor template. So here I show you an article which illustrates the collaborations between the GenScript with the Marsden laboratories from the University of California in San Francisco. So in this article, the author described that the use of the recombinant viral vectors to genetically reprogram the T cells for therapeutic purposes may have hindered due to its high off-target rate. And in this article, the author described that they had successfully developed a CRISPR-Cas9 genome targeting system that does not require the viral vectors, allowing the rapid and efficient insertions of a large DNA sequences, which greater than 1 kilobase at a specific site in the genomes of a primary human T cells, while preserving the cell viability and function. So the workflows on the right panels here illustrate the concept of the entire project. So in this project, the author will need to integrate this GFP tag that flanked with a long homology arms at both ends of the GFP into the housekeeping gene, which is known as the RAD11A gene, without the use of the viral vector in both CD4 plus and CD8 plus cell. So here is the workflow that the author adapted. So the workflow is very similar to the workflow that we use for CRISPR knockout. What the author did was to design a guide RNA of interest and also the DNA donor template. And then this guide RNA was mixed with the Cas9 endonuclease for 1 hour in order to form the RNP system. And while waiting, the author isolated the T cells and resuspended in the electroporation buffer. So after 1 hour, the newly formed of the RNP system was mixed with the DNA donor template for 10 to 30 seconds prior to the mix with the T cells in the electroporation buffer and further electroporate for 30 minutes. After that, followed by the cell expansion in the 96-well plate, and the GFP expressions was evaluated to investigate the successful of the CRISPR knock-in effect. So here is the data. The use of the RNP together with the double-strand DNA as a donor template show that GFP expressions at approximately 50% in both the CD4 plus and the CD8 plus cells. So the author next confirmed that this method can be broadly applied by targeting the sequences in different locations in the entire genome. So the author engineered the T cells by fusing the GFP into different genes, including the CD4, CLTA, ACTB, FBL, and TUBA1B. And then the GFP expressions was evaluated. And as you can see from this result, that a GFP expressions was observed ranging from 36% to 23% to each of these templates. So these results were further validated by using the confocal microscopy in order to confirm the fusion protein localizations in the endosome, plasma membrane, clathrin, actin, nucleolide, and microtubules. So while the use of the double-strand DNA as a donor template can achieve a high on-target rate, the study have shown that the use of this donor template can have caused the off-target rates across these templates. So this led to the author started to think the use of the ssDNA as a donor template. So here is a data that's studied by the author. So the results show that the use of the ssDNA could have achieved a similar, if not better, of the editing efficiency compared to the use of a double-strand DNA as a donor template. And the efficiency increased with the increased amount of the ssDNA was used. Furthermore, the author had also studied the cell viability, and the results showed that the number of the colonies that transfected using the ssDNA was better compared to the number of colonies that transfected using the double-strand DNA as a donor template. Additionally, while the ssDNA can achieve a similar on-target rate as the double-strand DNA, it could actually reduce the off-target rate much more better and efficient compared to the use of the double-strand DNA as a donor template. So here is a table that summarizes the advantages and disadvantages of these three different platforms. So as I mentioned, the use of the viral vectors to deliver the donor template is commonly used due to its high transductions and the transfection efficiency, but the off-target rates and the higher toxicity are the main concerns among the researchers. And while the use of the linear double-strand DNA may have achieved a high on-target rate, but the off-target rate and a high toxicity remain to be resolved. So in contrast, the single strand of the DNA may seem to be benefit compared to these two platforms due to its low off-target rates and low toxicity. But the only drawbacks of using the ssDNA is the production was difficult. So the reason that I say it was difficult to generate is because in the past the technology to synthesize a long ssDNA was not available, and therefore the productions of the ssDNA was through two different pathways. A, the use of the exonuclease in order to degrade one strand of the PCR product that was labeled with a phosphate group at the 5' end, or to go through a complicated IVT, RT, ssDNA workflow, in which the first step involved the in vitro transcriptions to generate an ssRNA from the double-strand DNA, followed by a reverse transcriptions to synthesize a RNA, a DNA hybrid. And lastly, it involves the incubations with the sodium hydroxide in order to degrade the RNA to produce a single-strand DNA. So fortunately, we have currently GenScript has the ability and the in-house technology to chemically synthesize a long nucleotides of the ssDNA easily. So here I would like to summarize what I have just mentioned before. The use of the RNP systems helps to accelerate and simplify the entire genome editing workflow. And then the synthesized sgRNA avoids the inductions of an innate response due to its absence of the 5' triphosphate groups. And then the ssDNA may seem to be the best option as a donor DNA template. So now I would like to move on to briefly discuss about some other extended products that offered by GenScript based on the CRISPR technology. I will talk about the solutions for gRNA library, the solution for CRISPR cell line. So what are gRNA libraries? The gRNA library allows you to synthesize the gRNA molecules that can mutate, activate, or repress every gene in the genome simultaneously. And it is ideal for large-scale screening, for knockout gene expression or transcriptionally activate the genes in the genome, and also an ideal alternative to short RNA-based screens that is prone to off-targeting and false negatives. Here I show you the workflow of how the gRNA library is constructed with our in-house facility. So what GenScript needs from the researchers is the gRNA design. Or GenScript also provided a pre-validated gRNA library for gene knockout, genome-wide knockout for the humans and the mouse species. So after we acquired the design, this gRNA oligo will be synthesized using our in-house semiconductor-based technology. So the quality of this gRNA oligo will be validated using the NGS to show that all the gRNA library has been constructed, followed by the gene cloning into the vector backbone that licensed from the Broad Institute. And then this followed by the library amplifications and the purifications using the plasmid midi-prep. Here I show you a classic QC examples of the gRNA library that was constructed in-house facility. So this gRNA library consists of more than 62,000 of the gRNA molecules. The quality of these gRNA molecules was validated using the NGS. So this table summarizes the NGS data. So the rank 10% indicates the 10% of the sequence that distributed on the left-hand side, and then the rank 90% indicating the molecules that distributed on the right. So it was considered as an equal distribution when the ratio between the 90% to 10% was less than 10. The smaller the number, the better it is in order to ensure maximum editing, the screening efficiency. And then the NGS data also show a total 100% coverage for the total gRNA oligos that are being constructed. So how can you utilize these CRISPR libraries after being generated? So the studies have shown that the screening using the CRISPR is of particular advantages due to its specificity, its versatility, and simplicity. So what the researchers need to do is after receiving the amplified library, the researchers can then package a pool of this gRNA library into the lentivirus in order to generate a pool of the knockout cell. And then by applying different treatments depending on your project, the researcher can perform either the positive screening and a negative screening. So this gRNA molecule can serve as a DNA barcode that can be used in order to do the cell counts that carrying this gRNA molecule by the high throughput sequencing. Here I show you a classic study conducted by the Shellamsen group in the year of 2014. So just a little bit of the background. The vemurafenib, the PRX, is a RAF inhibitor to treat the melanoma. And some patients do not respond to this regimen because the cancer cell gain the resistance by mutagenesis. So the objective of this project was through the gene target screening in order to identify the genes that suppressing the resistance to the BRAF inhibition. So what the author did was to order a genome-wide knockout library that consists of more than 60,000 of the gRNA molecules in order to target more than 18,000 of the genes. And this gRNA library molecules was packaged into the lentivirus in order to transfect the A375 cells. And again, by applying the PRX to perform a positive selection. The NGS was used in order for the bioinformatic analysis to validate the result as well as to identify the genes that suppress the resistance to BRAF inhibition. So here I show you the data. So as you can see that after applying or treated with the PRX, the number of colonies has greatly reduced. And then what the author did was to extract the genome from the cells that survive after the treatment for NGS validation. So through the NGS screening, the author managed to identify a couple of the top candidate genes after the analysis. And these genes include the NF1 and MET12, NF2, COL3, TALA2B, and TATA1. So now I want to move on to talk about the solutions for the CRISPR cell line. Here I show you the workflow of that adopted by GenScript in order to create a gene knockout or a gene knock-in cell line using the CRISPR method. So the entire workflow can be discussed in 5 different phases. The first phase involves the whole cell characterization. So depending on the project as well as the cell type, numerous tests will be carried out in order to guarantee the success of the project. And then the phase 2 involves the guide RNA design, the guide RNA delivery system selection, the guide RNA Cas9 synthesis. Now, this phase 2 can be conducted concurrently with the phase 1. And it is critical to select an appropriate guide RNA delivery system to make sure the success of this project. And this followed by the transfections of the guide RNA, the Cas9, with or without the donor template depending on the project to create the cell pool. So the enrichment of the transfected cells was carried out by FACS sorting or the antibiotic selection. Additionally, numerous tests will be carried out in order to guarantee the success in modifying the cell pool. So this phase is considered as a checkpoint. Therefore, from the test, there is a strict guideline that GenScript will need to follow in order to guarantee the success or the success in creating the modified cell line. And this followed by the single cell cloning, and the last phase was a genome typing or the validation using different methods such as the Sanger sequencing, the qPCR, Western blot, or the functional assay. So here I show you an internal and case study that GenScript did it for our pre-existing customer. So we know that the KRAS gene can encode the KRAS proteins, which is very important in the cell division. So the objective of this project was to knock out the KRAS locus in the human colon cancer cell line by creating the indels in the exon 4. So The guide RNA and the Cas protein encoded components were packaged into the lentivirus for transfection. And due to the absence of the DNA as a donor template, the repair was carried out using an error-prone non-homologous end-joining pathway. So a specific primer was designed to target the sites where the double-strand break was occurred for Sanger sequencing. So here I show you the Sanger sequencing data. So each of the clone was Sanger sequenced in order to select the homozygous mutations at the KRAS genes at exon 4. So this Sanger sequencing data of the parental cell shows a perfect sequencing data. So whereas in the clones where the indels was introduced, a missing sequence was found in this region. Additionally, the result showed that the result shows a homozygous mutations that could potentially lead to the loss of the KRAS expression. So to prove that, GenScript performed a Western blot analysis to confirm the loss of the expressions of KRAS KRAS protein in this particular clone. Okay, and now I would like to take this opportunity to mention about the stable cell line generations that GenScript is offering. So the entire workflow is similar to the workflow that generate the CRISPR knockout or the CRISPR knock-in. So the workflow can be divided into 5 phases as well. So the first phase involves the whole cell characterizations. And to generate a stable cell line, the researcher will need to design and we will help to synthesize the genes of interest and package into the viral vector. So for long-term expression, GenScript utilized the lentivirus for transfection. So the phase 3 involves the transfections of the lentivirus to create the stable cell line, and then followed by single cell cloning. And lastly, depending on the project, GenScript offers a several validation methods, including the FACS scan, Western blot, qPCR, and luciferase assay. So here is just a couple of the examples of the QC or the validation data that GenScript did for our pre-existing clients. So what I would like to highlight here is GenScript has a team of cell line experts that are ready to discuss with the clients or their project. Additionally, GenScript has an excellent track record in creating the stable cell line. So last but not least, I would like to put up this slide that shows the CRISPR services that offered by GenScript, including the CRISPR guide RNA plasmid, the ssRNA, the sgRNA, ssDNA, the CRISPR cell line guide RNA library, as well as the microbial editing services. So now I would like to stop here for my presentation today, and I am willing to take any questions from the floor. Thank you.